in these studies, and they are in line with, it goes from background exposure. You know, the green is where the University of Washington dentists were, and the line, the triangular lines above the bars represent background levels of exposure in various studies, and they're right in line in their dentists with background exposures ranging from the lower part of their study out to higher level exposure in some of the dentists. And what you see in their work is, again, a lack of a threshold that they're seeing effects down to very low urinary mercury levels across this population of dentists and dental assistants. So this kind of a data set, you know, if you don't want to take it by itself, is certainly supportive of thinking that it's going to be hard to find a threshold across a diverse population that's got lots of vulnerability factors and co-exposure to things like methylmercury from fish. In fact, methylmercury dose response has been looked at as a linear response by Axelrod et al. 2007, rather than as a threshold dose with a point of departure and an RFC type of approach. So you know, they did that work based upon the Faroe, Seychelles, and New Zealand studies to look more at cost benefit. You know, what is the benefit even from some kind of an industrial mitigation of mercury release at levels that might lead to less than the RFD or more than the RFD. Where are we on that continuous frame? But you know, their analysis clearly showed the likelihood for a linear response down to fairly low levels. Oken et al. 2005 and 2008 also shows at low levels of methylmercury exposure that you can find effects in the general population. So that I bring up these methylmercury data because number one, amalgam could be additive to these effects from methylmercury. This is occurring in the same people. And number two, mercury ion in the brain may well not have a demonstrable threshold, but there may be effects or at least increased risks at fairly low levels of exposure. It's what this is talking about. Okay, so the total mercury level in the brain is likely the key influence on neurodevelopmental risk. And so that if elemental dose from amalgam is below it, let's say that the elemental dose by itself that someone is getting from amalgam is below some threshold, it could still be above the threshold in that person if they have fish mercury, methylmercury. So if this is a hypothetical construct here, if brain mercury levels are on the x-axis and percent population affected on the y-axis, and the way I drew this is that there is a threshold somewhere below 5 parts per billion in the brain for a mercury effect on some neurological outcome. Well, so if amalgam exposures lead you to be less than 5, you might think, well, that's fine and dandy. However, if your level of methylmercury exposure has you out at a level of 7 or 10, and then you're already up the dose response curve, so that the amalgam exposure would not be in the subthreshold range anymore. You'd be in the linear part of the slope. And so that's really what we're looking at here. Here I put together data from animal studies as well as from human cadaver studies. So the dashed lines going vertically is the one on the left, or it looks sort of more in the middle of that plot, is the low end of the human brain cadaver mercury levels, and it's about 7 parts per billion. And the right side is in folks that had something like 10 to 15 amalgam surfaces. So you can see that that's the range, at least from Eggleston et al. 1987, that's the range of mercury in the brain that comes from amalgam, is the high end, and without amalgam, those folks that didn't have any amalgam fillings were on the left end. Probably that's coming from methylmercury. And superimposed on that are the swimming latency and rearing activity, neurotoxic effects of mercury in animal studies. And so you can see that they didn't find a threshold. Well, they didn't have enough data points probably to demonstrate a threshold, but at least they're showing effects in animal studies that are below the levels of effects of mercury in the brain that we're finding in humans. So there are some caveats to that analysis that I talked about in my homework, but at first take at least, it does appear that levels of mercury that we have in the human CNS, either from background from methyl mercury from fish, and then when added to that from amalgam exposure, are in a range where we're seeing effects in animal studies, just based upon levels in the brain. I'm going to skip this slide. It's right out of the NAS book, but I'm probably going to run out of time. So I want to get to how to model this and approaches that FDA could think about based upon the Silver Book and some other things. There's the concept of additivity to background disease, and this gets at one of the questions earlier today. Well, what does it mean if you're above the RFD? And I think what it means is, even if you're in a subclinical level of effect or a theoretical level of effect, it's what most likely is occurring is that people who might be at greater risk for Alzheimer's, Parkinson's disease, learning disorders, auditory aging-related auditory deficit, that their risk may be compounded by the elevated risk from inorganic mercury, from elemental mercury exposure from amalgam. If you're above that magic point of departure, the RFC, and perhaps even if you're not above the RFC, because I'm not sure we could find a threshold. So let's see. So does the RFC protect the perinatal period? That's question 1B. Does the threefold intrahuman uncertainty factor cover adults to perinatal difference and vulnerability? Well, certainly we've got some vulnerabilities in very early life that those occupational studies would not be covering, including the immaturity of the blood-brain barrier, which would allow greater access not only of elemental, which gets into the CNS pretty well, but any inorganic mercury that may be formed in red blood cells and might have an issue trying to get up to the CNS. Certainly that would occur more easily in the perinatal period. Immature detoxification, there's data that plasma glutathione levels are 10 times lower at birth than they are in more mature children and adults. And so who knows what that means about CNS glutathione levels, but certainly systemically glutathione appears to have an ontogeny that we need to be thinking about. And of course the critical window of brain. So those are more pharmacokinetic, toxicokinetic issues. And then there's the toxicodynamic issues of the critical window of brain development for things like methylmercury and lead, that we know the developing CNS is more vulnerable to those neurotoxicants, and likely also for elemental mercury. So my conclusion on this is the RFC, given this threefold intrahuman uncertainty factor, is unlikely to adequately account for perinatal vulnerability. What happens if you exceed the RFC? Where would effects start? Which was basically, I'm rewording question 2A. The amount of mercury in the CNS may already be at a subtle effect level in the human population. It's a similar concern that we would have for, say, background levels of PCBs, background levels of dioxin.
Doctor, you have 20 seconds.
20 seconds, okay. So obviously then, if we're already at a background level of concern in the general population, that any additional exposure may be seen to have some kind of an effect for the neurotoxic kinds of endpoints that I was talking about, at least an increased risk for those effects. So let me just see if I could get to, I talked about the negative epi studies. It's all in my homework, so hopefully you'll avail yourself of that. But just let me conclude saying amalgam represents an excellent example of conceptual model 1 of the Silver Book, where low-dose linear modeling is really warranted. That the threshold on an individual, there may be a threshold on an individual level, but the threshold on the population level may be very difficult to find because of the various additivities I have listed there. And at a minimum, I would say that FDA should either develop their own bright line for assessing risk, or the RFC should be, for mercury, should be reassessed by EPA.
Thank you very much.